Hello everyone, this is Manogya. I hope you all having a good day. So guys, today we are going to study a very important topic, that is Kelvin cycle. See, it is one of the most important as well as the easiest topic to, study. So now what is Kelvin cycle? See, Kelvin cycle is one pathway which is taking place in all the photosynthetic plants. Okay. Now, Kelvin cycle is broke down into three major steps. The first one is carboxylation, the second one is reduction, and the third one is regeneration. Now, what you feel when you heard the name carboxylation? It feels like it is related with carbon, right? So, what exactly takes place in carboxylation? It is the fixation of CO2 into a stable organic molecule, right? The fixation of CO2 into stable organic intermediate that is taking place in the carboxylation. And it is one of the most crucial steps of Kelvin cycle. The enzyme which is used for the fixation of CO2 molecule is Rubisco. Now, why? There's one interesting fact you'll get to know about Rubisco, that it has both carboxylase as well as oxygenase activity. That's why it is called as Rubisco. That is RUBP carboxylase oxygenase. Okay. Now the second step is reduction. See, reduction is not just a one-step process, it is a series of various reactions. Okay, that leads to the formation of glucose. That's why we have called it reduction. Now the steps. Now here one important thing that we have to remember is the steps involves the utilization of two molecules of ATP for the phosphorylation and two molecules of NADPH for the reduction of CO2 molecule. Okay, now coming to the third step, that is regeneration. Regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue. See, now we have formed glucose. Okay, now the product, we have got the product, right? Now we want RUBP, the enzyme which we are using, which we are using as intermediate. We want that thing back. So for the regeneration of RUBP, it is important. So here one molecule of ATP is utilized for the phosphorylation, okay, to form RUBP. See, this is a very brief and a very small introduction to a Kelvin cycle, and all the sentences that I have been taught you is taken from NCRT. Even the diagram is from NCRT. Okay, now we'll go in the depth of the cycle. This is the basic framework. Okay, this is the basic structure that I have built for you people. Now we'll go deep and we'll study what exactly and how things are taking place, right? So, see, now I want you to be very careful, okay, and please carefully listen to this. See, we have one RUBP molecule. The very first step, carboxylation. Now what we are doing is we are taking one molecule of RUBP and we are taking one molecule of CO2, right? And the enzyme we are using is Rubisco. And I told you that Rubisco has dual property. It can be used as oxygenase or it can perform carboxylation. So here we are taking CO2. In the presence of CO2, RUBP was is fixed with CO2. So now the Rubisco will work as carboxylase. And now what happens? It forms a molecule which is a bit unstable. So it broke down into two molecules of PGA. Okay. Now the very important factor to keep in mind while studying Kelvin cycle is we have to just simply remember the carbon number. What we have to do is we have to balance out the carbon number. Every time in every step you have to remember the carbon numbers. See, RUPP is a 5 carbon containing compound and CO2 has 1 carbon. So on the left side of the reaction there are total 6 carbon. So we have to balance the carbon, okay, and nothing else. On the right side, now if I am taking one molecule of RUBP and I am taking one molecule of CO2, okay, so what I should get? I should get 6 carbon, right? 5 from RUBP, 1 from CO2. So the total number of carbon that I should obtain is 6 carbon. So the two molecules of PGA are formed. See, PGA is 3 carbon containing compound. So we'll get two molecules of PGA. Okay, now it's very simple. Now if you once know how to use one RUBP and one CO2, you can easily study the reaction with see, when you will give the exam, you're not going to be asked for if there can be a question like for one RUBP and one CO2 molecule, you will get two molecules of PGA. So now for three molecules of RUBP and three molecules of CO2, how much PGA you will get, right? So it's very simple. If I'm taking three molecules of RUBP, RUBP is 5 carbon containing. So here 15 and three molecules of CO2. So total 18 carbons. So since we know that PGA is 3 carbon containing compound, so we should get 6 molecules. We can divide 18 by 3, we'll get 6. So 6 molecules of PGA will get. So now again, if I am taking 6 molecule of RUBP and 6 molecule of CO2, how much PGA I should get? I should get 12. And there's I'll tell you one easy trick to remember all this. See, if we are taking X amount of RUBP and we are taking X amount of CO2 molecules, we'll always get 2X amount of PJ. See, the number of RUBP and the number of CO2 is going to be same every time. Okay, so the amount of PJ that we'll get is twice the amount of PJ or twice the amount of RUBP or twice the amount of CO2. It's a very easy trick to remember. So if there's going to be any question in any exam or any statement can be given to you in assertion in reasoning, okay, like if I'm taking Y molecules of PGA and I have taken X molecules of CO2, how much RUBP I would have taken, or it can be like if I'll get Y molecules of PGA. So tell me how much molecules of RUBP I would have taken. So it's simple. We know that RUBP is twice the PGA which is generated is twice the number of RUBP. So if I'm taking Y molecules of PGA, then definitely I would have taken Y by 2 molecules of RUBP. If I'm taking 2 Y molecules of PGA, I must have taken Y molecules of RUBP. It seems like a bit complicated, but it's not that complicated. Okay. So now the way we'll study the cycle using 6 molecules. Okay, I'll tell you why I'm taking 6 molecules. You might have read the cycle using 3 molecules of RUBP, 1 molecule of RUBP. We'll study the cycle using 6 molecule of RUBP. So if I'm taking 6 molecules of RUBP here and I'm taking 6 molecules of CO2, definitely I'm getting twice the number of PGA. So I'll be getting 12 molecules of PGA, right? Now we should balance out the carbon numbers. See, RUBP is a 5 carbon content. I'm taking 6 molecules of it. 6 into 5, 30 carbons. And 6 carbon I'm taking total 36 carbon. So the PGA should have, and we know that PGA has 3 carbon. So 3 into 12, 36. Okay. Now the carbon is balanced. Now, as I've told you, that there are two important things which is taking place in the reduction phase. If you might have remembered, the first one was the phosphorylation, and for phosphorylation, we would have used ATP, right? Two molecules of ATP we used, and two molecules of NADPH we have used for single RUBP, right? So now, if I am taking one RUBP, for one RUBP, I am taking two molecules of ATP, right? So for six, how much ATP I should have used? Twice. So again, we are using 12 molecules of ATP. So in the very first step of the reduction, the phosphorylation steps take place. So the PGA molecule is being phosphorylated and it becomes 1,3 by PGA. Again, the carbon number is same, 36. Now, the second step is the reduction step. Okay, here NADPH is being converted into NADP and 1,3 by PGA is converted into PGAL. Okay, so again, these two are the very important steps of this Kelvin cycle. Okay, and both we have used 12 molecules of ATP and 12 molecules of NADPH, right? Now what we have got is 12 molecules of PGAL, right? Now, see, when you will study for any competitive exam, it is important. to just have a little basic knowledge of how exactly the glucose molecules, is formed. So that's why I'm teaching you this. So now what happens next is these 12 molecules of PGL will take 3 molecules of PGL. Okay, so I'm taking 3 molecules of PGL and it is 3 carbon containing. So 3 into 3, 9 carbons. And I'm also taking DHAP. See, DHAP and PGAL are interconvertible forms. Okay, these are the same things. Okay, both are 3 carbon containing. I can so what happens? 3 molecules of PGL and 3 molecules of NADPH are taken up. We have taken from these 12 molecules. So now we are left with 6 molecules of PGAL, right? I have taken 3 molecules of PGL and 3 molecules of DHAP from this 12 molecules. So I have taken 3 into 3, 9 carbon and 3 into 3, 9. Total I have taken 18 carbons. From 36 carbons, I have taken 18 carbon containing compound. Okay. So now these form 6 molecules of fructose 1,6 biphosphate. See, we know that glucose, fructose, galactose, these all are 6 carbon containing compounds, right? So I have to balance it with 18 carbon. And I know that fructose is a 6 carbon containing. So again 18 divided by 6, we'll get 3 molecules of fructose 1,6 biphosphate. Okay. From this fructose 1,6 biphosphate, one molecule of H3PO4 is being removed and 3 molecules of fructose 6-phosphate is formed. From these 3 molecules, one molecule of fructose 6-phosphate gets converted into the one molecule of glucose. Okay, that we are studying. See, why I have taught you the cycle using 6 RUBP molecules, because to make you understand that how exactly one molecule of glucose is formed. If I would have taken 3 molecules of RUBP or 1 molecules of RUBP, it will be a bit difficult for tell you that how exactly this carbon is formed. So now from 3 molecules of fructose, 1 molecule of fructose is converted into glucose. Okay, now we are left with 2 molecules of fructose. Now the process in which glucose molecule is formed, that comes in reduction phase. Now we'll study what we'll do with the left out fructose, 2 fructose molecules and 6 molecules of PGAL. From 12 we have taken 6 molecules. So we are left with 6 molecules of PGL and we are left with 2 molecules of fructose 6 2 molecules of fructose, right? That we'll study in the part regeneration. Okay. So now from these 2 molecules of fructose 6 phosphate that we are left with, I am taking, and we are left with 6 PGL. From these 6 PGL, I am taking 2 PGL molecules. Okay, so 2 into 3, I am taking 6 and 2 into 6, 12. Total 18 carbons. So these two combinedly forms 2 molecule of xylose 5 phosphate and 2 molecules of erythrose 4 phosphate. Xylose 5-phosphate is a 5 carbon containing compound and erythrose 4-phosphate is a 4 carbon containing compound. So 2 into 5 10 and 2 into 4 8. So here 18 carbon. The carbon is being balanced. Okay. Now see what we are going to do in regeneration is we want to regenerate RUBP, right? Because in the very first reaction we are using RUBP and we are fixing it with carbon, okay, and then we are forming our glucose molecule. So there are two concerns of Kelvin cycle. The first one is the formation of the glucose molecule that we have successfully done. Okay, we have get a glucose molecule, and the second most important thing is to get back RUBP. Okay, so now in regeneration, we'll study that how exactly we are getting RUBP back. So, uh, okay, now this xylose 5-phosphate is being formed. We get 2 molecules of xylose 5-phosphate. Okay. Now what we do in the next step, we'll keep this as it is and we'll take 2 molecules of erythrose 4-phosphate. Okay. So now these 2 molecules of erythrose 4-phosphate, we are taking 2 molecules of DHAP. So 2 into 3, 6 and 2, 4, 8. So again, it is a 14 carbon. Okay, 14 carbon here. So what we'll get is, we get pseudoheptulose 1,7 biphosphate. Pseudoheptulose is a 7 carbon containing compound. So we'll get 2 molecules of it. So again, 2 into 7, 14. So the carbon is being balanced. Okay. So from 2 molecules of erythrose, we will get 2 molecules of pseudoheptulose. And see, from this 6 PGL that we are left with, I have utilized 2 molecules of PGL here and 2 molecules of DHAP here. So now I am left with 2 molecules of PGL. Now what we'll do is, 2 molecules of pseudoheptulose 1,7 biphosphate, 1 molecule of H3PO4 is removed. So it becomes pseudoheptulose 7-phosphate. So we'll add up here 2 molecules of PGL, which we are left with, okay, and 2 into 3, 6, and here's 14. So it is a 20 carbon containing compound, which will form 4 molecules of xylose 5-phosphate and 2 molecules of ribulose 5-phosphate. See, 4 into 5, we know that xylose is a 5 carbon containing, okay. So it is a 4 into 5, it is 20. So why I'm writing here 2 molecules of ribulose 5-phosphate is, these 2 molecules comes from here. So in total what we are getting, 4. See, I'll repeat for you people. In this step we got 2 molecules of xylose, okay, and in this last step what I've got is, I got 2 molecules of xylose from this pseudoheptulose and PGL. What I'm getting is 2 molecules of xylose and 2 molecules of rubisco. So here I am getting 2 molecules of each, and since here I have got 2 molecules of xylose, so I'm adding up that here. So total we are getting 6 molecules, right? The xylose 5-phosphate is again an it gets converted like just how fructose molecule gets converted into glucose, because both are 6 carbon containing, and so here ultimately we'll, just like that, xylose gets converted into ribulose, and so we get 6 molecules of ribulose 5-phosphate. And in the very last step, see, you have to keep in mind that I've told you for one RUBP we'll take one ATP molecule for its regeneration. So in the very last step you will see that for 6 molecules of ribulose 5-phosphate, here 6 ATP molecules are being utilized, and then it gets converted into RUBP, 6 molecules of RUBP. So since we are starting our cycle with 6 molecules of RUBP, we have to, we have to get back our 6 RUBPs back, right? So now in the very end step, we have got our 6 RUBP molecules. Okay. So this is what your Kelvin cycle was. I'm guessing it is a bit tricky, but it's very easy if you're going to balance out the valencies of carbon. You have to just balance out carbon on both the sides. See, you don't have to remember all the steps in the reaction, because I'm pretty much sure it is a bit tricky to remember all the steps. But why I have taught you everything in detail, because just to give you a brief outlook that how exactly your glucose molecule is formed and how exactly your RUBP molecule is regenerated. So you have to just keep in mind the basic things, that in carboxylation we are using Rubisco, okay, and we are getting 2 molecules of PGL in the very first step. This is the most crucial steps. Then again in reduction you have to keep in mind that if I'm using 1 RUBP, I'm using 2 ATP molecules and 2 NADPH. If I'm using 6, so I'm using 12 ATP and 12 NADPH. These are the 2 important steps of this reduction phase and how we are getting glucose, okay. And in regeneration, what one thing you have to keep in mind is that in the end, in the very last step, we are using 6 molecules of ATP. See, if I'm taking 1 RUBP, I'll use 1 ATP. If I'm getting 2 RUBP, then I'll use 2 ATP. Here I'm getting back 6 RUBP molecules, so I'm using 6 ATP. Okay. So I hope you had a good time. You have a good time reading this. Once you will revise it, you will get to understand it. It's not that difficult. So take a very good take a very good take care. Okay.